Hi, good afternoon, all. So I think we can start. It's already 12:34, so we have waited long. So good afternoon to all of you who have joined. So we'll try to, yeah,
Good afternoon, sir.
yeah, yeah.
Good afternoon, sir.
Afternoon. Yeah. Kindly mute yourself because there's from some of you there's a lot of background noise actually. So I would suggest to mute. So we'll start where we left yesterday. I mean, it got disconnected a bit abruptly actually, and then there was some minor net issues over here, so I just couldn't connect back. Anyways, so this is where we were yesterday. I mean, I've given you a glimpse of what is tissue culture, and then a brief history of tissue culture, some of the major landmarks achieved with respect to the progress of tissue culture from 1902 all the way to the 1960s 1970s when the media was when the MS media came up. So these are some of the major landmarks, and then we also talked about the different types of tissue culture that is being practiced, or that is practiced as per the experimental requirements. So which included the callus culture, cell suspension culture, protoplast culture, anther culture, and we were on to meristem culture. So as I mentioned yesterday, that in meristem culture, we generally don't look to develop a callus. Actually, we take the meristematic tip from the stem and we culture that, and this is generally, and then we like it leads to the propagation or leads to development of an entire new plant. And this because we are using shoot meristem tip or shoot apex. This is generally a practice that is used in horticulture for mass propagation of clones for commercial market. So, and also for developing virus for the production of virus free plants. We'll come to that later, like why we use meristem culture for virus free plants. But just to give you a brief that because the meristem tips are generally considered they are virus free, they are not affected by the virus infestation or contamination. So that's why when we want to of a virus infected plant, when we want to have a virus free plant, we just do a meristem culture. We take the shoot tip and we start the meristem culture, and that leads to the development of a virus free plant. And this is especially used in horticulture for especially for the plant species that are not grown in native conditions. I gave the example that if you bring up a plant species which is accustomed or acclimatized to the cold conditions of the north, and then you try to bring it in the say in the area of Rajasthan, it won't grow. But using the meristem culture under controlled conditions of light, temperature, and humidity, we can actually I mean with proper optimization, we can actually lead to a point where we can multiply the plant. Okay, so moving on from meristem culture, there was another terminology that has been listed in the syllabus, which is somatic embryogenesis. So I thought I'll just give a brief of that and we'll move on to the next aspect actually. So talking about somatic embryogenesis, I think the name itself suggests that it is the embryogenesis taking from the somatic cells. Now in nature, embryogenesis is restricted to ovules and involves the fusion of male and female gametes. We are aware of that. But there have been several cases where non-gametic cells or non-germline cells have been found
So
to
much
develop
breaking.
embryo. Hello? Hello?
The voice is breaking, sir. Hello.
Um, I will try. I think there's some, I mean, the internet sometimes is not at its best actually, but do let me know wherever it's breaking. I'll try to repeat that part. All right. So in,
Okay, sir. Okay.
yeah. So in some cases, the non-gametic cells or the non-germline cells have also been found to undergo embryogenesis. But what is more important, that even in such cases, the embryos are actually developed inside the embryo sac or the female gametophyte. So, I mean, this is the coming out from the somatic cells or the non-germline cells. This is a fertilization independent embryogenesis. And tissue culture have actually exploited this aspect of non-fertilization independent embryogenesis to the very best. And it has shown that irrespective of whatever the ploidy level or whatever the specialization of the plant, they are capable of forming typical embryos, and that can germinate. So this embryos coming from the somatic cells are known as somatic embryos, and the entire process is known as somatic embryogenesis. I mean, I listed it here because this actually forms a key component of tissue culture studies. Now, this, I mean, the slides so far, or what we discussed yesterday, is actually to give you a holistic view or a glimpse of the tissue culture, different techniques or different aspects of tissue culture. And then what I'm moving on to the nutrient media constituents. This is, I mean, as most of you said that you have done a course on plant tissue culture, you have done practical. So I'm aware that majority of you might still be, or might well be aware of it. But as I'm trying to go in a sequential manner, I'm just covering things like one after another. So talking about the nutrient media constituents, this is also an important pillar of the tissue culture, like all the different techniques or the totipotency we talked about somatic embryogenesis. They all form integral and critical components of tissue culture technique. And so is the nutrient media constituents. We are quite well aware that the mineral elements that are required, some are required in larger quantities, which include calcium, magnesium, nitrogen, phosphorus, sulfur. So we call them macronutrients or major elements or macro elements. Now, apart from these other mineral elements such as iron, manganese, copper, zinc, boron, and molybdenum, they are required in small quantities, and hence they are known as micronutrients or microelements. Now, I also mentioned as part of the progress of plant tissue culture, people tried different kinds of media preparations, different kinds of constituents or components in the media, among which one of them were the vitamin components, vitamin B. So, and I also indicated that this is quite, I mean, since the time of P.R. White in the early 1930s, this is quite well established. Even today, we use vitamin solutions as one of the constituents when we prepare media in the lab. And that's what I've mentioned here, that in addition to all these inorganic constituents that are listed above, organic constituents such as vitamins and amino acids, in some cases, we do use amino acids also. They are also essential components of nutrient media that we use for tissue culture purposes. Now, vitamins, I have already mentioned the thiamine, riboflavin, pyridoxine. These are the ones. I have pulled out a list of the stock concentrations, stock solutions, or stock concentrations or components that is present in the MS media. And I mean, this is a 20x solution composition, but we prepare the stock solutions in the lab and then we dilute it accordingly and we use it. So this is exactly the way we handle it in the lab actually. Unless we are using commercial ones, I'll just show you actually. Yeah. I hope it is visible to all. Yes.
Yes, sir.
Yeah.
Yes, sir.
So this is how it is commercially available. This is how we purchase it. It comes in glass bottles, also plastic bottles, but this is how we purchase it. And if you can read it, it is written Murashige Scoop medium with calcium chloride and vitamins, without sucrose and agar. So it's and you get, I mean, different combinations. We get with vitamins, we get without vitamins, we get with vitamins and sucrose, we get without vitamins, but plus sucrose. So we get different combinations. It's just that what, I mean, it based on our experimental requirements, what do we need? So this is how it comes actually. But this is the commercially available form of Murashige Skoog. As you know, there is a plant tissue culture practical also. So in that, we ask the students to prepare MS Media by mixing all the components. We don't provide them the commercial one. That's part of the training. Actually, we ask the students to prepare the different stocks and mix them in the right proportion to make the MS Media, because as part of the plant tissue culture, we should be aware of what are the different components, how they are prepared, and how we mix them to make the final MS Media. So that's why in the practical, like, unfortunately, I don't think so you guys will be here soon. But otherwise, when we do the tissue culture experiments, or when we do the tissue culture practicals, we teach the students to do this. So like, say we split the class into 4 different groups. So one will prepare a stock of all the macronutrients, which will cover this. One will prepare the stock of all the micronutrients. Now, among these micronutrients, iron is not added. Iron is not added. We prepare a separate stock for FeEDTA. We provide it in the chelated form, so to allow its slow release into the medium, actually. So we don't use just FeSO4. We give it as the FeEDTA, actually, so that it is in the chelated form.
Sir, your voice is breaking.
All right, hang on then. Just a second, I'll try to connect with my phone, actually, and see if that makes a difference. Okay, so is it better now? Hello.
Earlier one was better, sir. Earlier one was better.
Is better or not? I mean, it's not good enough.
Your mic off.
Yeah. Sorry for this. Yeah. So what I was mentioning is we allow the students, ask the students to make the different stocks, and we don't give iron as such. We make a separate stock for iron EDTA. And then we have a stock for the vitamin solution, 200x stock for the vitamin solution, where we add the different vitamins. Now, apart from vitamins, in some cases, amino acids are also used. Some of the amino acids are also used, actually. And glycine is one of the most commonly used amino acid that we used in the nutrient media. Other include glutamine and asparagine, but I mean, that are not that commonly used. Glycine is most commonly used, and these amino acids, they serve as the organic source of nitrogen. But the nitrogenous salts that we are using here, especially ammonium nitrate and KNO3, potassium nitrate, they provide the bulk of the nitrogen required. You can see they are providing the maximum. I mean, they are supplied in the maximum amount, and they provide the bulk of the nitrogen required for the plants. Apart from all this, we also add as a carbon source, which is sucrose. When I showed you the packet, it showed without sucrose and agar. So sucrose is one of the carbon sources that we provide, roughly between 2 to 5%. About 3% is what we use in the lab. So these are the different components or different constituents that are used in media preparation. And this is a list of the role of the different components. I'll not go with through all, but I mean, just I mean, I think you guys might be aware with majority of it. What is the requirement of potassium? What is the requirement of magnesium? It is required for chlorophyll molecule. Calcium is required for cell. It is a constituent of cell wall. Nitrogen is required for amino acid synthesis and so on and so forth. And then you have the micronutrients, the zinc, molybdenum. These are they act as cofactors for some of the enzymes. Then we have the vitamins, thiamine and ascorbic acid, and myoinositol. These are the three major ones that are used, not all the amino acids. So we generally make mix 3 or 4. That is the most commonly used ones. And then, of course, sucrose, as I mentioned earlier, it serves as a carbon and energy source, as well as it acts as an osmotic agent. So you can go through them in detail. These are the the role of the different constituents that we add. Each one has have a specific purpose.
Sir, can you please go to previous slide?
Yeah. Tell me.
My connection broke, so sorry. Okay, this is the
Yeah, this is the previous slide. I was talking about the what are the different constituents we prepare when we prepare the MS media from stock solutions rather than using a commercial one. We use the prepare a stock for macronutrients, we prepare a stock for micronutrients, we prepare a stock for iron EDTA, and we prepare a stock for organic nutrients, which is mainly which mainly includes vitamins and glycine if we are adding any amino acid. And then
okay.
we keep all these stocks in fridge, and whenever we need it, we mix them in the required proportion. We add sucrose and we add agar and we autoclave the media. I'll come to that actually. I'll come to that. So, and then coming on to this slide, this is a list of the role of the different components that we add in the media, because each one have a specific purpose. And as you have seen in the history of tissue culture, that they have been as the tissue culture progressed, different kind of components or different kinds of ingredients have been included in the media to see its response or to see its effect on the plant growth. And that's how the Murashige school came up with this composition, because they realized that each mineral or each compound has its own specific requirement. Okay. Now, apart from the inorganic mineral nutrients, your vitamins, the amino acids, the plant growth regulators, as you might be aware or as you might be knowing, plays a very, very important and critical role actually. And although all these explants that we introduce into the tissue culture for callus formation, they already have some endogenous level of the growth hormones. But it is more often than not, I think, we always add these growth hormones or these growth regulators. You know, these different hormones. I guess you are aware of auxins, cytokinins, gibberellins, abscisic acid, all these different growth hormones.
Yes, sir.
Okay, good. So we also call them plant growth regulators, because they are not only, I mean, they are hormones by chemical nature, but they are acting as plant growth regulators. So we call them plant growth regulators. So what I was trying to get at, that although these explants have certain level of endogenous growth hormones or growth regulators, it is more often than not, it is necessary to exogenously provide this into the nutrient media. Even though they are required in very, very minute quantities, like in the micromolar or nanomolar range, but they are still required, and they play very vital roles in tissue culture, conditions or tissue culture growth actually. Now, one of the major ones, as you guys might have read before, as you guys might have been aware, one of the major ones are the auxins. Now, auxins are known to be promoting root growth, but that happens at lower concentrations. At higher concentrations, it induces callus formation. Now, this is also an outcome of the history that we were discussing yesterday, that people have tried different concentrations and see and like observe this response on the callus formation or on the plant growth under tissue culture conditions. And then they realize that at lower concentrations, it favors root initiation, whereas at higher concentrations, it induces callus formation, because it induces cell division and or embryogenic differentiation. So that's why it induces callus formation. Now, what is interesting that when we take the explant, I mean, we do this in the lab actually. So when we take the explant and we want the callus to be like we want the explant to form callus out of it, we give high concentrations of auxin and cytokinin. Now, but once the callus purpose is sorted out, and I mean it is started resulting into a plantlet actually, or shoot. Then we want the shoot to start developing roots so that it can start the newer shoot that has come out of callus. We want to start developing root so that it can start extracting nutrient from the media that we are providing. So then we transfer it to another medium where we are providing low concentrations of auxin, and that causes the rooting to initiate actually. So this is how during the tissue culture conditions, we play with the different concentrations of the growth regulators or the growth hormones as per our requirement. We initially grow them at higher concentrations, put the explants at higher concentrations and a specified ratio of auxin and cytokinin so that the callus formation takes place. And then once the purpose is sorted, we bring it back to lower concentrations of auxin so that the root initiation takes place. Now, the most commonly used auxins in the tissue culture conditions are IAA, which is indole acetic acid, indole butyric acid, and naphthalene acetic acid. And some other includes 2,4-D and this PCPA, this para-chlorophenoxyacetic acid is not very commonly used. We generally play with IAA, IBA, and NA. I mean, sometimes 2,4-D, but quite rarely actually. And among these, indole acetic acid and butyric acid are the naturally occurring auxins, whereas the others are synthetic ones. Now, the same thing which I talked about auxins, they also applies to cytokinins. Now, cytokinin, we all know, plays a role in cell division and differentiation, especially of the adventitious shoots. So it also leads to, at higher concentration, it also leads to callus formation because it's involved in cell division. So what we do at the initial stage, as I mentioned, we use a specific ratio of auxin and cytokinin to develop callus from the explants. And then at a later stage, when we have transfected the callus with agrobacterium, we'll come to that later when we talk about transgenics and all. So when we have infected the callus with agrobacterium, and we want it to start generating shoot, we bring the callus back to lower concentrations of cytokinin so that it starts giving shoot. So we name the media, like whatever kind of media we are using, we name it accordingly. One is called a pre-culturing media, the callus inducing media, or the regeneration media. So we name it accordingly actually. So you might see different nomenclatures for such media, but they all have like slight variations in the auxin cytokinin concentration, slight variations in the vitamin stock vitamin concentrations, but the basis MS as the basis remains the same actually. Now, the commonly used cytokinins are kinetin and BAP, benzyl aminopurine. These 2-IP, zeatin, and adenine are not that 2-IP and zeatin are not that commonly used. We mainly use kinetin and BAP actually. Now, as I mentioned, gibberellins, auxins and cytokinins are the two most commonly used. In some cases, gibberellins, ethylene, and abscisic acid are also used, but very, very rarely. In fact, to be honest, I have never used gibberellins, ethylene, so abscisic acid as far as the tissue culture thing is concerned. I have never done that actually. So, but in some cases, because say you are growing the plantlet, you want it to grow faster, and as we know, gibberellin leads to internode elongation. Gibberellins, we do include gibberellins in the tissue culture media. Apart from these inorganic, like the MS media, vitamins, amino acids, plant growth regulators, in some cases under specific requirements, people do use some additional supplements. And one of the most common ones is polyamines. The polyamines include pitricine, spermidine, and spermine. They promote organogenesis and somatic embryogenesis. So just to enhance the process, polyamines are used. And in some cases, people do use activated charcoal, but activated charcoal's main purpose is to absorb the toxic exudates from culture tissue. Now, talking about toxic exudates, I would like to mention here that when you, I don't know if you guys have come across this, those who have done tissue culture experiments, but when you leave the your explants or you leave your callus on a particular media, tissue culture media for very long, and you don't do subculturing. We generally do subculturing wherein we prepare fresh media and transfer the plantlets or callus to the new culture bottles, to the new media. So that is called subculturing. So when we don't do subculturing and leave it for very long time, you will start seeing a browning of the media. You start seeing your media, which is generally transparent or whitish in nature, it starts turning brown. So that is basically because these toxic exudates are being released by the plants and the callus growing on that media into the media. And that's why it starts turning brown actually. So that browning, we need to avoid that actually. But in some cases, if it is happening, then we include, then people do include activated charcoal to absorb these toxic exudates. Now, of all these, apart from all these important nutrients or important nutrients and vitamins and plant hormones, we need especially when we are not doing cell suspension culture, we are doing the callus culture or these kind of things, we need a solid agent, actually solidifying agent, and that is called as gelling agent. Now, one of the prime reasons of using solidifying agent is also if you are not doing cell suspension culture. So if you're doing a callus culture and or protoplast culture or the anther culture, those kind of things, if you have a static liquid media, this will submerge, and because of lack of oxygen, it will die. So we need a solidifying agent, we need a solid media so that they can be placed on the surface and can be allowed to grow actually. So those components are called gelling agents. Of course, I know the agar is one of the most commonly used gelling agents, but these are some of the properties listed here for gelling agents, that it should be inert, it should not affect the composition of your media. It's not like that it should be releasing some kind of nutrients or some kind of minerals in there. It should be completely inert. It should be able to withstand sterilization conditions, because we all know we need highly aseptic conditions for tissue culture purposes. Everything has to be super sterile. And we need to withstand, so the gelling agent that we are using, it should withstand the sterilization condition. It should not break down or disintegrate during autoclaving. It should remain. So this is another important property that should be taken care. It should be liquid when hot, and it should be solid when cold. I mean, for obvious reasons, so that we can prepare the media, pour it, and once it cools down, it solidifies actually. And the semi-solid nature of the medium enables the plant to be placed on its surface. That's what I mentioned actually, that it should not submerge down. It should be resting on the surface, all these explants or calluses. So these are the major properties of a gelling agent. And agar is one of the most commonly used gelling agent. But for tissue culture purposes, for when we move on to a slightly advanced stages of tissue culture, rather than using agar, we use Gelrite or Phytogel. This is also similar to agar. I mean, it is a gelling agent, but the slight difference between agar and Gelrite, that agar is slightly yellowish in color. So when we prepare the media, when you pour the media in the bottles, culture bottles, it gives a slightly turbid kind of appearance. Whereas Gelrite or Phytogel is very clean, is transparent actually. So what it helps actually, it helps in a proper observation. Say if there are any kind of contamination developing, or their roots are not growing properly. So if you have a transparent kind of media, clear media, it will help you. It will help you to observe properly and then act accordingly. Whereas agar is slightly translucent or turbid in color actually. If you give me a second, I'll show you. I have some bottles here. Can you see this bottle actually?
Yes, sir.
Yeah.
Yes.
So this is how a plant tissue culture bottle looks like. Just a second. So can you see this? So this is what Gelrite looks like, and I don't know if it is visible on media. Can you see these roots over here?
Yes, sir.
Yes, sir.
Yeah. Yeah, can you see these? So this is gel, right? Actually, this is an old media, but if when it is fresh, it is a bit more transparent actually. So you can clearly see the roots in here actually. And this you won't see when I mean, if you use agar as the gelling agent. So this is the difference between Gelrite and agar. And this is done by some of the MSc students, actually MSc first year students. So we were doing some tomato tissue culture thing. So this is done by them. Thankfully it was there lying in the lab. So I thought of showing it to you guys. So I mean, this is what I wanted to share about the gelling agents. Then coming to
So-
these, yeah.
Sir, can you once again repeat the importance of polyamines? Actually,
Polyamines is mainly required for promoting organogenesis and somatic embryogenesis actually. So it is not used in all the cases, like we generally don't use it, but say for example, in some of the tricky situations or in some plant tissue culture conditions where the plant is not propagating or it's not forming, there are no organ development taking place, organogenesis is not happening or embryogenesis is not happening. In that we try to, this is part of the optimization, like we don't do it normally, but these are additional supplements, and as per our requirements, say you do a couple of times and you see that no organ formation is taking place, you don't see shoot coming up, or you don't see embryogenesis happening, so then we'll kind of try to optimize the media, and we add a pinch of or some particular concentration of polyamine to see if that makes a difference. So this is like part of the optimization process, but this was just for your information that these are some of the additional nutrients that are being added, that are being used as and when the requirement arises.
Okay,
All right.
sir.
Okay. So moving on, one of the key aspects or one of the key factors is the pH requirements. How many of you know? Okay, let me rephrase the question. Do you know what is the pH of human body?
Slightly alkaline. 7.4.
What is it? What is it? Do you know?
7.4.
7.44.
Okay, good. And what is the pH of plant sap?
Sir, around 5 to
6.
5.2.
Yeah. 5.8 to 6.
You have read that before, is it? I mean,
Yes, sir.
okay, good. So not many people know actually. That's why I asked, but it's good that you guys know. So it is correct. The pH of the plant sap is 5.8. Is around 5.7, 5.8 actually. And that is one of the prime reasons why pH makes adjusting the pH of the media that we prepare is so important. Is because when we prepare the media, we want the pH to be somewhere around 5.7 to 5.8. This will help the plant to grow actually, otherwise it will be in a completely different environment. So because when we make the pH of the media very close to the plant sap pH around 5.7 or 5. It influences the uptake of nutrients, and the solubility of salts, gelling efficiency also is affected. So for keeping all these things in view, we try to maintain the pH around 5.7, 5.8. So when we make the media, before adding the agar, always remember, before adding the agar and also before adding the sucrose, we don't add sucrose and agar. We first prepare the MS media with all the components and everything, and then we adjust the pH. to 5.7, 5.8, and then we